Welcome back to the online class, students. In the previous class, we were discussing about the urine formation, where the blood has been filtered in the glomerulus and the filtrate will be formed in the lumen of the Bowman's capsule. And today's class, we are going to discuss with the functions of the tubules. Okay, functions of the tubules we are going to discuss. So the renal tubule has been differentiated into proximal convoluted tubule, Henle's loop, and distal convoluted tubule. So the first thing we are going to discuss about proximal convoluted tubule. Okay. So the first function is we are going to discuss about proximal convoluted tubule. Okay. So the proximal convoluted tubule, as we have discussed already about the nephron. So this is a glomerulus, Bowman's capsule part, and this is the PCT, that is proximal convoluted tubule, and this is the U-shaped structure is the Henle's loop, and this is the DCT or distal convoluted tubule, and this part is the collecting tract. Okay. So now coming to the functions of proximal convoluted tubule. So this proximal convoluted tubule has been lined by cuboidal brush-bordered epithelium. Okay. So it is lined by what? So cuboidal brush-bordered epithelium. Brush-bordered epithelium. What is a cuboidal brush-bordered epithelium? So the cube-shaped epithelial cells which is provided with the finger-like projections. So by having these finger-like projections are brush-like projections, so which provides them more area for reabsorption. Okay, and PCT is a region where maximum amount of essential nutrients has been reabsorbed. Okay, the first function is, so the essential nutrients are absorbed in the PCT region. Not only that, 70 to 80% of water as well as electrolytes are also absorbed in the PCT region. Okay, so 70 to 80% of water as well as what? Electrolytes. Different types of electrolytes has been reabsorbed in the PCT region. And along with that, even you can see that HCO3 minus, that is bicarbonate ions, are also reabsorbed. Okay, so that is a major function of PCT. Then not only the reabsorption, even you can see that the selective secretion of the tubule. Okay, the selective secretion. So preferably hydrogen, potassium ions, and even ammonia also secreted into the tubule. Okay, so secretion of what? Hydrogen ions you can find. So potassium ions you can find, and even ammonia. Ammonia also has been secreted into the filter. So which helps to maintain the pH and ionic balance inside the filtrate. Okay. So that is a major function of proximal convoluted tubule. The second major part of renal tubule is Henle's loop. That is Henle's loop. As we know that in the Henle's loop, this is the Henle's loop where you can find, so this is a descending limb and this is a ascending limb. Okay. Now the filtrate from proximal convoluted tubule enters into the Henle's loop. So Henle's loop is a region where minimum reabsorption takes place. Okay, but so the major function of Henle's loop is to maintain high osmolarity inside the medullary interstitium. As we know that Henle's loop preferably found inside the medulla. So Henle's loop maintains what? High osmolarity. Osmolarity inside the medullary interstitium. That is a major function of Henle's loop. How does high osmolarity has been maintained? So when the filtrate enters into the descending limb, so this descending limb is permeable to water. So it is permeable to water, so that when the filtrate it moves to the descending limb, it will become highly concentrated. Then from the descending limb, when the filtrate enters into the ascending limb of enlaced loop. In the ascending limb of enlaced loop, you will find the nature that so it is permeable to the electrolytes and almost impermeable to the water. So that when the filtrate move to the ascending limb, so it will become diluted. Okay, so that is a major function of the enlaced loop. Then the filtrate from the ascending limb, when it will enter into the DCT. This is a distal convoluted tubule. So in the distal convoluted tubule, the third point and the third region is the distal convoluted tubule, where you can find conditional reabsorption. Okay, so what you can find there? So conditional reabsorption, that is the required amount of reabsorption of preferably water and sodium ions takes place. Along with that, even you can see that bicarbonate ions are also reabsorbed in the DCT region. And in the DCT region, even so selective secretion, so that is addition to the filtrate. So selective secretion of potassium ions, hydrogen ions and even ammonia into the filtrate takes place. So by the addition of this secretions, so which help to maintain a particular pH or ionic balance of preferably body fluids. And even we can say that, so it helps to maintain potassium and sodium concentration in the blood. Okay, so that is a function of distal convoluted tubule. So then the filtrate, later it enters into the collecting duct. So collecting duct is a large duct which is extending from, so this part what I mentioned is a cortex region and this part is a medulla region. So as you can see that collecting duct is extending from the cortex region into the medulla region. When the filtrate enters into the collecting duct, so here maximum reabsorption of water takes place. Okay, and little amount of urea from the collecting duct so will be reabsorbed into the medullary fluid. So this is a medulla region, so it will be reabsorbed into the medullary fluid. So in this way, the concentrated urine has been formed. Okay, so the next thing is mechanism of. So the next topic here is mechanism of concentration filtrate. The next one what we are going to discuss is mechanism of concentration filtrate. So that is how the concentrated urine has been formed, which is the mechanism followed there. Okay, so mechanism of concentration filtrate. Mechanism of what? Concentration filtrate. Okay. Now, so what is a mechanism? So in case of mammals, you can see that a concentrated urine has been formed. Otherwise, you can mention it as hypertonic urine has been formed in case of all the mammals. Okay. All the mammals you can find what? Concentrated urine has been formed. And this is also one of the adaptation to conserve the water. So this has been, so here the major role of Henle's loop is very important. And even I have mentioned about vasa recta. Okay, so the mechanism we are going to study the role of Henle's loop as well as vasa recta. What is vasa recta? So already we have discussed that. So the efferent arteriole emerging from the glomerulus, so which runs parallel to the Henle's loop, we call it as vasa recta. Okay, so Henle's loop and vasa recta plays a very important role. That is the fluid inside the Henle's loop. That is in the descending limb which is running downwards, in the ascending limb it moves upward. Okay, so they are running in a opposite direction. Okay, in the Henle's loop you can see that the filtrate run in a opposite direction. Okay, and the next thing in case of vasa recta, so as I told that, so the vasa recta is what? It is a peritubular capillary running parallel to what? Vasa recta, we call it as, Henle's loop, we call it as vasa recta. Then, so as it is a blood capillary, so it will contain what? Blood. Okay. So, the blood here also, it flows in the opposite direction. So, here, it flows in this limb or in this region, it flows downwards. In this region of vasa recta, it flows upward. That is in the opposite direction. And not only that, so here the proximity. So, this is the ascending limb of Henle's loop. So, it is moving downward. This is the descending part. Descending limb of what? Vasa recta. Okay, that is. So, the proximities are opposite. And this side you can find that here is a descending limb of the Henle's loop and this side is the ascending limb or ascending region of vasa recta is found. That is, the proximities are also in the opposite direction. Okay. So, which helps in maintaining high osmomolarity. So, that is high osmomolarity means in the cortex region, you will find around 300 moles of osmomolarity per liter. And in case of medulla, you will find around 1200 moles of osmomolarity per liter. Okay. So, this is also not only with the help of Henle's loop as well as vasa recta. So, even sodium chloride and urea also plays a very important role. That is, sodium chloride and urea plays a very important role. What is the role here? So, that is the sodium chloride, NaCl, found in the ascending limb of Henle's loop will be exchanged with it enters into the descending limb of vasa recta. Later, so it flows to the descending limb and later enters into the ascending limb or ascending region of vasa recta and it is released into where? Medulla region or medullary interstitium or medullary fluid. So, in that way, it helps in increasing the osmomolarity. Okay. The next is urea. A small amount of urea, so enters into the thin segment of ascending limb. Okay. And this urea, along with the filtrate, flows into DCT. From the DCT, it enters where? Into the collecting duct. From the collecting duct, later, again, the urea is released back to where? Medulla region. So, it helps in maintaining high osmomolarity. So, here, you can see that the Henle's loop and vasa recta has been arranged in a specified manner that we call it as counter current mechanism. What we call it as counter current mechanism, which helps in producing highly concentrated urine. And not only that, even it helps in easy reabsorption of water from collecting duct, so that the urine formed is highly concentrated in case of mammals. That is around, so the urine formed at the end is around 4 times highly concentrated than the urine formed in the initial times. Okay. So, this is regarding the mechanism of concentration filtrate. So, let us see some of the questions. So, the first question is, mammals produce a urine which is option A hypotonic, B hypertonic, C isotonic, D all of the above. It is highly concentrated hypertonic. B is the answer. In which part of the renal tubule 70 to 80% of water and electrolytes are reabsorbed? PCT, DCT, Henle's loop, collecting tissue. It is in the where? It is in case of PCT. Then, concentration gradient is mainly caused by hydrogen ions, sodium chloride, urea, sodium chloride and urea. The answer is D, sodium chloride and urea. The next question. High osmomolarity or countercurrent mechanism is caused by flow of filtrate in opposite limbs of Henle's, okay, and flow of blood in the vasa recta in opposite direction, proximity of Henle's loop and the vasa recta, all of the above. So the answer is all of the above. Mammals are characterized by metanephric kidneys with a loop of Henle, mesonephric kidneys with Henle's loop, metanephric kidneys without Henle's loop, pronephric without Henle's loop. As we know that, so mammals are highly advanced. They'll be having metanephric kidneys, and you can see the presence of Henle's loop. The answer is A. So next one, PCT is lined by columnar epithelium, brush-bordered columnar, C. Cuboidal epithelium, cuboidal brush-bordered epithelium. The answer is cuboidal brush-bordered epithelium. That is regarding today's class, students. Thank you.